Now, I'm going to put up my first disclaimer. I'm not an ethicist, okay? So if you do have any questions, I'm more than happy to take the questions back. But what I'm going to talk about now is really how we're harmonising the structure and putting in place a structure for an international iPSC banking with regards to consent forms and access user agreements. So the overview that I'm going to give is really concentrated on the second point, the EBiSC, the governance designed for accessibility, take you through some of the consent deposition, access and use agreements, and then describe some of the challenges and strategies that we've actually put in place to really understand this very dynamic process. So IMI and EBiSC is really an opportunity to harmonise and bring together these very fragmented iPSC cell research and banking in US and Europe. And really we feel that it's an excellent time to consolidate and strengthen the EU's bioscience base to really avoid duplicating effort and to create this high-value iPSC resource that will be available to all. So the vision of EBiSC is really to bring together all of the knowledge, the cell lines, the expertise together in this one place and really build a world-class research-grade IPS line bank. We acknowledge that it's going to be derived from a diverse and special donor cohorts, but with, hopefully age-matched controls that will be of value to the researchers moving forward. And it needs to be accessible for research use and for anybody to use moving forward. We've touched on some of the donor awareness and protection and QC that we've put in place within EBiSC, and I'm going to describe some of the harmonised international standards and templates that we're actually generating. And just to reaffirm that we're moving forward, we anticipate this will be a sustainable and not-for-profit bank. So we have a number, if you're if you come to us saying we would like you to assist us in making an IPS line, we do have a number of donor information and consent forms that have been generated to allow the thorough sort of basically agreements to be put in place to allow research use of the lines moving forward. There's a material deposit agreement, an AUA, an access and use agreement, an EAUA, and a participant access and use agreement, a PAUA. So there's a whole raft of forms, but we help you if you contact us to walk you through those particular steps. As an organisation, any line coming in has to be agreed through the consortia board to actually allow money to be directed towards that particular IPS line derivation. And there's another access committee, an ethics advisory board, which there are two of our scientific advisory board members actually present in the audience today. So it's actually a very cohesive group and consortia moving forward. So for the donor consent, the key features is that consent is vital. It respects the donors and enables the research use moving on. It's a voluntary informed donation, so there's no money associated with it, and we actually generate the iPSC derivation with a limited participation and withdrawal capability moving forward. We support multiple projects and long-term access and use so that it's a sustainable resource moving forward. And we actually, there are multiple access agreements that can be put in place moving forward to actually utilise those cells. The consent, is actually, we have the ability of looking at DNA and genetic information, as Laura has actually described, but there are no immediate benefits, financial results or health associated with the donation. And we also ask that there's a recontact option present in the consent form. For the material transfer agreements, the key features is we're banking iPSC lines, not primary tissue, and we've really harmonised the deposit and use provisions throughout the consortia to minimise the barriers to accessing these cells. EBiSC provides the repository, the QC, as Glyn has outlined. The depositor retains ownership of the cell line, and the depositor is free to collaborate and use those cell lines that we generate in their research. There's no downstream IP or royalties on the research outcomes, as these cells can be sent to FPIR partners for commercial use, etc., with an agreement in place. So the MTAs for use, access and use agreement, the user license to use the banked lines for research use, no therapeutic use is stressed. The existing lines that come with encumbrances, the non-EBiSC consent and third party, these are actually the hot start lines that I described. They will be transparent in the user agreements associated that you can actually download from the information that Laura actually showed. The user pays for access and not-for-profit fee structure, and EBiSC gets no IP rights or royalties associated with it. We have a standing access committee and an ethics advisory board, and we also have, sort of acknowledgement of the depositor. It's actually, we can put, as Laura showed, the publications associated and more information as it's generated. We minimise the user hurdles, and we've actually chosen lines that are sought by researchers that are consented appropriately and that are unencumbered. So what we're building is a sustainable economic model. We actually really value the ongoing contribution of participant resources and expertise, and we understand the interests and motivations to facilitate tissue donations and deposition, and we're really engaging with networks of individuals in these disease areas. But what we really want is feedback and make this a living document to actually change it, the consent and the MTA frameworks, to suit the researchers with the maturation of the EBiSC project. So the challenges that we faced are really the internal coordination, the public-private partnerships, the harmonisation, and this has actually been quite an uphill struggle, as shown by the sort of cartoon here. We now have established a broad consent policy and ethical, legal plurality. We have an implementation policy, so showing the provenance of existing lines, and we have really engaged with multiple stakeholders with very diverse interests. We understand to some extent the property and the IP, who owns the cells, so we have that very clearly stated. And it's only for research use, the donors, depositors and users that are interested in these lines. We have a very large data storage and management system that underpins the information associated with these lines that is accessible, and we're really building the sustainability and not-profit operations in a commercial environment.